Welcome back, everyone, to the IFIN's annual meeting. My name is Kathy Greaves, and I'm the Global Nutrition Scientist for the Plant-Based Protein Platform Group here at the Kellogg Company. We're very excited for this session focusing on an in vitro method for determining protein digestibility. This topic coincides with the mission of the IFIN's Protein Committee, which is to inform future dietary protein recommendations by taking into consideration intake amounts and quality in a North American food supply that is undergoing a shift in the balance of animal, plant, and novel protein sources. I'd also like to welcome Dr. Jim House. Dr. House, welcome, and please introduce yourself.
Hi, my name is Jim House. I'm a professor in the Department of Food and Human Nutritional Sciences in the University of Manitoba in Winnipeg, Manitoba, Canada. And it's my pleasure to be here to talk about substantiating protein content claims. Are there alternatives to bioassays?
Thank you, Jim. As we get started, can you tell us a little about how you became involved in protein quality research and why this topic is of interest to you?
Sure, thanks, Kathy. I started, my PhD was in the area of amino acid nutrition and metabolism, and I've been doing research in this area for a number of years, and this sort of coincides with my disclosure slide here, just highlighting that I have support from a number of entities on both the animal and plant protein sectors, as well as serving on the board of trustees for IFINS and the board of directors for the Canadian Nutrition Society. But when getting back to the question of the specific research, I really became interested in the protein content claims when I was first approached by a novel, what was then a novel protein source. It was hemp, when they were seeking information on how to substantiate a content claim for food products that included hemp protein in Canada and the United States. And so we did what was called a protein digestibility corrected amino acid score. And this just sort of sets the stage for a little bit of background that I'd like to present to the group to highlight this whole issue around protein quality claims. So just to make sure everybody's on the same page, I'll just give a little bit of an overview of how we make protein quality and content claim statements on food products. So in the United States, the basis for protein content claims is both protein quality and quantity, and they use what's called the protein digestibility corrected amino acid score. In Canada, we use the protein quality and quantity as well as the basis for content claims, but we use a different system called the protein rating system, which is based on the protein efficiency ratio. Both of those official methods require the use of an animal bioassay. In this case, they use a rodent bioassay to determine the actual numbers. In other jurisdictions, there isn't a requirement for substantiation of quality, it's only quantity. And there is a proposed method also from the FAO that also looks at the issue of protein quality, and it's now called the digestible indispensable amino acid score, or the DIAS method. And this method also requires the use of a bioassay, and there's actually a preference for what species, human being first, and then pig, and then rat. But in essence, when we're talking about protein quality, we're really talking about two components: digestibility and availability, as well as the amino acid composition relative to the reference pattern. For the sake of today, I just wanted to give a little bit of an overview of the issues surrounding the measurement of that one component, which is really the digestibility piece. So if we're focusing on the PDCAS method, it uses something called the true fecal protein digestibility, which is a rat bioassay, and you're essentially measuring protein in and protein out. In this case, we're really looking at nitrogen in versus nitrogen out, and with a correction for what we call endogenous losses. And these are the official methods that are established within the Code of Federal Regulations within the United States. And they're based on the published methods in Protein Quality Evaluation Report 51 of the FAO, and also substantiated by some official methods of analysis of AOAC International. The other method that I mentioned, the DIAS method, looks at true ileal amino acid digestibility. It's not officially established, but there is a preference for using human, then pig, then rat. But there's been a lot of work recently trying to develop data sets using ileal digestibility values from pigs. But if we focus on the PDCAS, which is really pertinent to today's discussion, what we're really looking at is how we measure true fecal protein digestibility. It does require the use of a rodent bioassay, but increasingly food manufacturers are demanding evidence that ingredients have not been tested on animals. And so even if the ingredient company doesn't have that as a stipulation for their own manufacturing processes, if they're supplying to a company that's looking to create novel food products, there is oftentimes that requirement being placed on procurement that the products not be tested on animals. And this is a major challenge for suppliers of protein ingredients, particularly new sources of protein ingredients where there isn't existing data on protein digestibility. So how do you provide evidence of protein quality to support content claims for marketing messages to consumer, while at the same time meeting that need of not having those products being tested on animals? And so really, are there other options that we should be looking at? And so I think one option that could be considered is focusing on what we call in vitro digestibility methods. And there are a number of these methods that have been approached. For example, there are commercial methods that have already been positioned by some labs, but the FAO, the document that actually substantiates the PDCAS method that's currently used within the United States, actually did position two in vitro methods within their documentation, one called the pH drop method, one called the pH stat method. They're both what we call static methods, but they've not been approved by an accrediting body such as AOAC or AOCS. So I think that's one of the challenges moving forward. It's the fact that we don't have accreditation for these methods. There are other models that have been positioned, including the InfoGest model, which is gaining interest in terms of looking at overall gastrointestinal digestibility. And other methods have been used recently to advance our understanding of plant protein quality. And just to give some examples from our laboratory, here's a work that we've looked at the pH drop method, which is a method that was cited within the FAO document, and very good agreement with the same samples that where we determined the in vivo PDCAS. And this is a variety of plant-based protein sources. And so we looked at either the pH drop method or a two-step method to look at gastrointestinal digestibility. And so both of these models do show promise as alternatives to in vivo digestibility measures. So that really leads to the opportunity today to further explore the potential for in vitro models for estimating protein quality, and some work that's been co-led by Elaine Kroll and myself, looking at this as on behalf of the protein subcommittee of IFINS, and really to establish a protocol designed to lead to a consensus approach for the validation of in vitro methods. And so this is really at the heart of the interest that I have in protein quality. It stemmed from an initial study done almost 12 years ago now, looking at protein quality of a single product, to now exploring opportunities to advance understanding in the world of protein quality. So Kathy, happy to engage in further dialogue on some specific questions that we could explore this topic.
Great. Thank you, Jim. That brings us to the topic for today. We're going to try a new format. So I'll be asking Dr. House a series of targeted questions for about 20 minutes. After that, we will welcome questions and discussion from panelists who are joining us. Before asking the questions, I want to clarify that the context in which all of these questions are being asked is related to the general population, not medical foods, nor the sole source nutrition, such as for infants, meal replacements, or complementary feeding. So to get started, our first question is, how important is protein quality for ensuring adequate protein nutriture among susceptible individuals living in North America?
Great question, Kathy. I think this is an opportunity for us to explore really how we use protein data in the existing nutrient databases all of our information about protein intake in the population, both in Canada and the United States and globally for that matter, really comes from what we call crude protein measurements. So all our intake data in the national database is all based on crude protein. So there's no correction for protein quality. It's only recently where we've been seeing attempts to sort of assess how protein quality really can have an impact on our interpretation of protein status within the population, in particular with subgroups. And so really it's to answer this question is a little bit challenging because we really haven't done the work yet to see whether or not protein quality matters. I think theoretically we can clearly state that protein quality does matter because we see it globally in certain populations that if you have inadequate balancing of protein sources, then you have susceptibility for stunting and inadequate protein nutriture. So I think at the North American level, I think it's probably more critical for subgroups of the population, in particular for growing children and for an aging population, where we're starting to learn more about the importance of quality protein for alleviating such conditions as sarcopenic muscle loss, and because that has implications for quality of life, in particular for our senior population.
Great, thank you. Our second question, with new and novel protein sources becoming more prevalent and with a shift in protein food choices by the consumer, is it important to consider protein quality when formulating foods that replace high quality sources?
Another great question, which has a number of potential answers to it. I think as you can see from my opening comments, there's alternative approaches to looking at protein quality, from just looking at protein content through to the assessment of protein quality by using the PDCAS method, which is what the focus for today is. And I think the challenge is that we have data in certain circumstances for some limited protein sources, but with the introduction of new flours, new isolates, new concentrates, new ways of blending these particular products, new thermal processing methods, all of these have an impact on both two of those components, both the digestibility and the amino acid composition of the resulting products. And so both of those components are really critical to have an understanding of how the final protein content and quality of the product should be positioned to the consumer so that they can make informed choices about the quality protein sources within those products. For example, we do see some protein sources that are coming up that could be extremely limiting in one amino acid or absent in one amino acid, which essentially means that by definition, they have a protein quality of 0 if they're missing one particular essential amino or indispensable amino acid. And so we need to be mindful to make sure that we've got processes in place that can ensure that we can guide new product formulations and ensure that we have quality protein ingredients to position to consumers.
Great. So since we know that protein quality depends on both the essential amino acid content as well as protein digestibility, considering that protein digestibility is currently used to calculate protein quality in North America for use on food labels, how important is digestibility compared to the essential amino acid content?
Right. I think it's a very interesting question because certainly the amino acid composition can be measured just chemically. You can use a quality lab service to determine amino acid composition, and it provides the amino acid score, which is one critical component for establishing PDCAS. Digestibility has oftentimes not been necessarily considered on a lot of these novel food products, and I think when we look at the effect of processing, in particular on digestibility, you can see dramatic differences on the extent and the type of thermal processing steps that it influences digestibility. So to go through a number of factors, there's a whole host of factors that influence digestibility, from presence of anti-nutritive factors or components within the food product that might limit digestibility. These oftentimes can be diminished by heat treatment, but if you go too high in your heat treatment, then you can actually significantly reduce digestibility as well by creating indigestible protein conjugates within the food supply. So it's it is still a critical component because we've seen the variability as affected by processing, and because of the interest in using protein concentrates and isolates and the ways in which they're being used in positioning new food products, I think it is important that there is a measure of digestibility that can take into account these particular components. And it can be challenging if you're focusing on using the current methods of in vivo digestibility, just simply from a feasibility standpoint and the number of samples that would have to be you would have to consider.
Great. So with that said, can you briefly describe what the gold standard measure is right now for protein digestibility?
Well, I can take the current definition of what the requirement is, and that's the true fecal protein digestibility for measuring PDCAS. That is the required method. Is it the best method? Well, I think that's why the FAO went through the process of looking at alternative methods and why they positioned the DIAS method, the digestible indispensable amino acid score. So in that method, it's the same conceptually as PDCAS, but instead of looking at nitrogen in and nitrogen out, which has some challenges because you can have modifying effect of the gut microbiome on the nitrogen content, and it can influence digestibility coefficients. The DIAS method uses ileal amino acid digestibility, and so that's a better reflection of how amino acids are taken up and absorbed. And amino acids are really the nutrients that we're concerned about from a human nutrition standpoint. The DIAS method is using technologies and techniques that have been around for a number of years, in particular for establishing ileal digestibility coefficients for use in livestock feeding scenarios. So that same knowledge has come forward into positioning that method. So ileal digestibility does present an opportunity to increase our knowledge of amino acid absorption, but the pig model has its own challenges. If we're thinking about animal testing, which is the heart of the next question, you're still talking about challenges if you're looking at the DIAS method because it's also a bioassay. There are some that argue that a human sample would be the gold standard because you're truly measuring digestibility within a human context, and actually humans can give informed consents, and so you're removing that ethical issue. But then you have the challenge of just the sheer logistical issues of measuring digestibility within human subjects. It's very challenging and costly and really limiting in the number of samples that you can theoretically do within that methodology.
Thank you. So given the importance of trying to eliminate animal testing at this point, what are the feasible options for protein digestibility testing, and how does in vitro digestibility testing compare to the gold standard?
Right. So looking at the feasibility, I think in my opening remarks, I showed a couple of examples of how in vitro testing using existing methods that were positioned within the FAO document, the pH stat and the pH drop methods could be used and readily established within labs globally. There's requirements to standardize the process, but it's an approachable assay, and it can be readily inputted in both testing labs as well as in an industrial setting as well. So I think those are feasible methods because then it opens up that methodology if it's done correctly and in an approved and validated approach can actually guide decision making with respect to product formulation. We've seen excellent agreement with the in vivo PDCAS for a number of different protein samples, both animal and plant-based proteins. But still there's a requirement for standardization of the approaches. And it's not just for nitrogen, if you're thinking about PDCAS, but if there is a move globally to adopt the DIAS method, then I think there would need to be much more work looking at the in vitro ileal amino acid digestibility assessment, and so that you can model amino acid digestibility at the ileum. And I think that's something that would be appropriate for for further examination of the DIAS method moving forward, because I see the use of a pig model or a human model is the same challenges exist for as we're talking about for PDCAS, and that it requires the use of a bioassay.
Thank you, Jim. Considering the complexity of the situation with two different potential models, DIAS and PDCAS, as well as a few digestibility tests, which in vitro digestibility test do you recommend as a standard method for use by food formulators? And what is your rationale for this recommendation?
Well, I'll put a bit of a caveat here. I think it's a little bit too early to make a sufficient recommendation, but I'll just give you from our own experience as well as a review of the literature that currently the methods that are receiving a lot of interest right now are the pH drop method and the pH stat method, and there's advantages to either, but the pH stat method is a fairly good method to use. It was established within the FAO, and it became very close to being an officially accredited methodology, but it just didn't quite get to that position. But certainly that method, because of the ease of implementing the test, as well as its accessibility, and the fact that it gives data for PDCAS that is comparable to the in vivo data, I think those methods, those simple static methods work well. Now, we've also seen you can get better agreement with in vivo data if you enhance the complexity of that model through what's called measuring what we call the gastrointestinal digestibility. And that's where the InfoGest model, that's a model that's been validated in a number of labs. And that's standardizing the conditions for multi-compartment in vitro digestibility testing. So you're truly looking at mimicking gastric digestion and intestinal digestion. So it's a better linkage to what's happening from a physiological standpoint. The challenge with the pH stat and the pH drop methods, they're good and robust methods, but the one criticism is that they're a mono-compartment method. So they're really just, all of the enzymes are added at once. And so that you're looking at digestibility. But even with that being said, I think it always comes down to is how good does it need to be? And so oftentimes when we look at the testing methods, it's a question of, okay, what is the risk that could be imparted by adopting this method? When we know that, yes, there could be variability in the answer that you get with relative to the accuracy or even the precision of a particular method. But when you think about plant proteins, for example, and all of the sources of variability that exist, theoretically in influencing both digestibility and amino acid composition, it's important that we keep that in mind when we're ascribing a potentially new test to look at protein quality within the for substantiation of protein content claims in particular.
Great. So knowing that we still have some questions to be answered around the methodology that we might be using, are there specific foods, and what are those foods that you would recommend be tested in an interlab test to establish a standardized method for public use?
Right. I think certainly one would be ideally it would be to use samples for which we have existing quality in vivo protein digestibility. So in the case of PDCAS, where you have good quality data on true fecal protein digestibility. And then you could look at establishing the protein digestibility on those samples, provided that the samples that you were using in your ring test or your interlab test is using the same processing conditions that were used to establish the in vivo data. Ideally, it would be good to do the in vivo data at the same time, but that's proving to be more and more problematic moving forward. But we do have a fairly robust data set now on in vivo true fecal protein digestibility measures. And so you could look at, for example, those samples where we have animal proteins. And so I would say casein is something that should be done irrespective of the ring test, because it's the one that's often used as a control in almost all of our tests. And then you could consider a couple of animal protein sources that have quality in vivo data, skim milk powder or a beef product or egg or whole egg or egg white proteins. And then a number of proteins that are from plant-based sources, including the flours, the isolates and concentrates, because we do have in vivo data for those. And I would recommend including both pulses, other legumes, and cereals in that mix as well.
Great. So then what are the next steps to achieve a standardized method for in vitro digestibility that would inform food formulation decision making?
Well, I think it's maintaining discussions and dialogue going forward with the regulatory bodies, but also to also be proactive. And that's really, I think, the goal of the protein subcommittee is to look at developing a protocol that could be positioned to establish a ring-tested method for validating an in vitro test, such as the pH drop or the InfoGest model, so that you can position a model to the as a valid alternative to looking at in vivo testing. And I think that's one of the things that I would recommend, is that it's not a single method that be considered. I think you want to look at making sure that we have opportunities to consider multiple methods. As I mentioned, there are commercial methods that are out there, but I think we'd want to look at how well opportunities and options. The same options may not be suitable for all individuals, but I think at the end of the day, it's a question of how well that data that's derived matches with the in vivo data.
Great. So we know individuals following a vegan diet are looking for high quality proteins. And we also know that the vegan associations will not certify an ingredient as vegan if animals are used in any capacity. How do you recommend that an ingredient or food company move forward with labeling regulations if in vivo digestibility measures can't be used?
Yeah, it's challenging, and I think this is something that needs really strong consideration about how one moves forward if you don't have existing data. Well, first thing to do is try to mine the existing databases, and we're publishing, and many other labs are publishing data on true fecal protein digestibility, but it's not a method that is widely used much in a research, at least in research settings. So I think there does need to be a gathering of all of the data that's currently available, so that we can see what exists and how to guide industry so that they know where to go to look at data that is readily available in terms of in vivo true fecal protein digestibility measures. Alternatively, there could be approaches where you are focusing on on data that are coming from another ingredient that's similar, but I think this is really a question for the companies themselves, and a discussion with the regulatory authorities about how one gets through this challenge about making claims in the absence of specific data, and what leeway there would be in using published data on a specific product that might be positioned for protein content claim. It is challenging.
Yes, it is. Thank you so much, Jim, for the information that you've provided to us today. That now brings us to a broader stakeholder discussion segment of this session. We will have two stakeholders on webcams to answer questions and discuss the session, as well as a couple of stakeholders who will submit questions that I will pose to the group for deliberation. First, I'd like to introduce Dr. Elaine Krull. Could you introduce yourself and tell us about your interest in this topic?
Yes, thank you, Kathy. I'm currently working as an independent consulting after retiring from DuPont Nutrition and Health, where I was a senior research fellow. I've had experience in academia as well as in pharmaceutical industry as well. But in my position, in those positions, I oversaw human clinical trials and animal studies, and focusing more recently on the role of soy protein and the health benefits conferred. Protein quality was obviously an important aspect of this work, and I was involved in calculations of PD-CAS, which required outsourcing the in vivo rat digestibility studies because there's very few labs that do that well. And then having conducted animal studies myself and knowing the requirements and when it's necessary to do animal studies, I understood the limitations and possible advantages of using an in vivo assay for nutrition labeling. And based on the extensive work that's been reported, not only by Dr. House's lab, but by many labs, if you look for in vitro digestibility, there's a lot of data that exists today. And I hold the position that in vitro assays yield comparable data to animal digestibility assays, and it significantly reduces the cost and time required. In vitro digestibility assays, in my opinion, will make it possible to test large numbers of food and food forms, which is impossible, as Dr. House alluded to, with the animal digestibility assay. So I'm currently, as you know, working with Dr. House on the IFINS protein committee, and my disclaimers were shown.
Thank you, Elaine. And Maya Villeneuve, could you also introduce yourself?
Yes, my name is Maya Villeneuve. I'm the Associate Director in the Bureau of Nutritional Sciences. And our interest in the discussion is related to a broad initiative we have in Canada called the Regulatory Modernization. It's an initiative by our Treasury Board Secretariat to update some of our, I would say, aging regulation and modernize them. And one of those initiatives is addressing official methods. A lot of our official methods are in regulations, and some of them are old because changing regulations take time. And one of the aspects is to take those methods outside of the regulations, which they can be updated more frequently based on the evolving science. And unfortunately, that initiative was a little bit delayed because of COVID-19. But what we did last December is we've accepted our official method was PR, and we've officially now accepted BD-CAS to measure protein quality on an interim policy basis. So, of course, right now we're requesting that in vivo data be used, but recognizing the field is evolving very rapidly, and this is why when the regulations are changed and amended, we intend to put a lot of our methods interpreted by reference. So there are types of regulations. So if there's new information that comes available, we can then update those methods more in a, I would say, easier fashion.
Great, thank you, Maya. So now we'd like to open this up for discussion. A note to the audience, we will take your questions after this panel discussion. So please go ahead and type your questions into the chat box at any time. So who would like to kick off this with our first question or comment?
Well, I'd be happy to start the discussion. Given that in the USA and Canada, the actual nutrient content for protein, the label allows you to be within 20% of the label claim for protein. So given that, and since Canada and the US are the only countries requiring protein digestibility studies for nutrition labeling of protein, what are the perceived downsides? And I think Dr. House kind of addressed it, but what would be the perceived downsides in Canada and the US if protein digestibility is conducted using in vitro versus in vivo assays? I don't know, Dr. House, if, given like there's already variability allowed on the label, and certainly your data showing the comparability of the in vivo versus the in vitro sort of would seem to fall, given that there's inherent variability in many of the other measures that go into that value.
Well, maybe I could provide a comment there. Certainly, when you're looking at variability, there's multiple sources when you're looking at digestibility and amino acid composition. You've got the environmental factors, especially for any crop plant-based protein sources. You've got environmental factors, you've got genetic factors, you've got processing factors, management factors, the effect of blending. They all of these create opportunities for variability. Even when you're looking at your sort of pea protein, you could have tremendous variation in the composition unless you're looking at specifically identity preserved pea sources, for example, where you have greater control on the environmental conditions. And so I think any of this discussion should recognize that it's against the backdrop of tremendous variability within the food supply. And so a discussion of the accuracy and precision of an in vitro method as an alternative to an in vivo model should be viewed in that context. And I guess the other argument that one could make is also in looking at an evolution of regulatory testing and what we've seen, for example, in the cosmetic sector, and how we've moved tremendously away from the use of animal bioassays, and yet it's still a requirement for nutrient testing. And I would argue that it's the only nutrient for which we need a bioassay for determining a content claim on a label. It's an important nutrient. I think we all recognize that historically we needed to ensure that the composition of that protein and the digestibility and availability of those amino acids was maintained, especially earlier on when processing steps were not quite as evolved, and we didn't have the knowledge of the effect of processing and sourcing on digestibility and amino acid composition. But I think an in vitro model, such as an in vitro PDCAS method, allows you to not only maintain that evaluation of amino acid composition, which is critical, but also to look at what factors can influence digestibility.
Maybe I can comment here, maybe just for clarification. In Canada, to label for protein, there's not a need for protein quality. It's only when you make a content claim that then you need to have basically the protein quality. There's other aspect as well when you're dealing with infant formulas and things like that, and some other categories where you need to provide protein quality. So I think I would want to make that clarification there, that it's not always required to label protein content. One question I do have for Dr. House, and we've had discussions over the years, and I'm sure he's going to recognize that question. There's a lot of methods out there. There's some I know you've mentioned that you've used in your lab and seem to produce good data. But often as a regulator, we're looking at something that's a little bit broader, that's used broadly, has been validated, so it's comparable, it's reproducible, and so on. Is there a method that seems to be further along to reaching those step of validation so that it can be recognized broadly by a number of recognized body like the FAO, for example?
Yeah, thanks, man. It's a great question. I think it's when you look back at the publications that were mentioned in the FAO document from 1991 on PDCAS, both the pH stat and the pH drop methods were actually going through, it almost seemed like they were going through a validation process, but they didn't quite take it across the finish line. That was back in 1991. So both of those methods are very well established. And in fact, if you look at publications recently, there's a number of groups that are looking at the pH drop and pH stat method to get a better understanding of digestibility in plant-based sources, and also new novel alternative-based sources that are coming from insects or culture systems or what have you, where we're generating information on for novel proteins. But I would argue that those methods are probably the farthest along. There may have been a gap of a few decades in their evolution, but the one that's also received a lot of interest recently is the InfoGest model. The only issue with that model, it's a model of overall human digestion, and so it's good for carotenoids, it's good for lipids, it's good for carbohydrates. It really talks about the digestive piece, and it doesn't really talk about the further analytical piece. And so you would need to invest the efforts into looking at either nitrogen or amino acid measurements and the validity of that method as it relates specifically to nitrogen or amino acid digestibility using the InfoGest model. But that method has gone through a ring test at least on the digestion elements of it, not necessarily on the analytical piece as it relates to protein quality.
So now I have a question from the chat, and it's a two-part question here, question comment. Thank you very much for your excellent presentation and discussion, Dr. House. In terms of the variability, since we know that there is so much variability, especially in pulses, is there concern that it could introduce even more variability that could render the amount declared less meaningful if we introduce regulatory methods that have significant variability? And go ahead with that question.
No, I think that's part of the validation process. And so I think the method itself is likely the reason that we're seeing variability between labs right now is because everybody has their own little ways of tweaking the methods. And I think, and accessing the enzymes, for example, that are needed for these particular in vitro methods. And so a really critical element to any advancing is the ability to standardize and have this method validated against existing in vivo data. So I think the digestibility piece is one element. Although we're not a primary focus for today, I think it's equally of concern to make sure that we have a validated amino acid and analytical system as well. While we do have validated methods for measuring amino acids, there are differences in the ways that people report amino acids. And so I think we do need to be ensure that people are reporting their amino acid content on the same basis. And so that's another element to measuring protein quality in the PDCAS system. But I think, yes, there is variability, but I don't think that we would put the if we have a validated method that's showing that we're showing that we have good reproducibility, that is accurate, that I don't think it would be putting the population any more at risk than if we're using an in vivo model for measuring digestibility.
Great, thank you. And to just add a bit of US context to this, there was a comment in the chat that the amount of protein declared on the label in the US is also not required to be adjusted for protein quality. However, the daily value when declared must be calculated based on the amount that is corrected. So that's just when we declare protein on the label in the US.
Right, and that would be true for saying whether it's a good source or an excellent source, you need to have that protein quality assessment.
And this might be a loaded question for Maya, but certainly when you look at percent daily value, Canada doesn't have a percent daily value for protein. Is that something that has been under discussion? I'm not sure if you wanted to comment on that.
Yeah, it was something that was looked at when we updated the nutrition labeling regulations in 2016, and we got quite a bit of comments and feedback on this topic. In the end, it was decided not to go ahead, but that's something, as you know, with time we get feedback from people, and we always take into consideration a lot of the nutrition labeling updates that were done in 2016 came from comments and feedback we got through the years, but also through our consultation, our official consultation process. So as more and more information is available, that's a way that we can update the regulation, especially when it comes to the table of daily value. The good news is it's now incorporated by reference. So it's we don't need to go through the full regulatory process to update those. Those are what we call more administrative updates, and they go only through our minister. They don't need to go through the entire regulatory process. So it's much quicker to update. So if there's a change, that can be done eventually, quickly.
Great.
this is a question maybe from me to Maya. The goal number one in the Canadian Council on Animal Care Strategic Plan for 2021 to 2026 is to do the 3 R's, the reduce, refine, replace, for animal studies. Is that part of your ongoing effort to modernize, to actually consider where animal studies can be replaced?
A lot of the aspects around modernization, they take into consideration, it's really taking into consideration every aspect as science evolve, but also if there is innovation, so new innovation as well. That was part of the plan for regulatory modernization. For sure, we still need to do our own work. We still need to make sure that the approaches, the methods we use are validated and they provide the information that needs to be made available so that we can ensure the safety and the efficacy of products. But that's why a lot of those aspects that are evolving with the science have been taken out of regulation, or the plan is to take them out of regulation into something that can be updated, much quicker than it was previously done. Just to give you an idea, the PR method had been there for a number of years, and a lot of scientific bodies like the FAO and other regulators were using PDCAS. Unfortunately, we were not able to update the regulation, but that opportunity is there now. And with taking those methods outside of the regulations, it's certainly going to allow for a quicker update. As Dr. House mentioned, DAS is also another one that's been discussing, and we know that that may be one that comes in the future as well. So we want to have that ability to update those methods rapidly as well.
Yeah, I think that's a really important comment around the DS method. Is while our focus today is on PDCAS, the FAO has proposed the DIAS method. It's being looked at by Codex. I think we shouldn't lose sight that there's an important opportunity to consider not only in vitro protein digestibility, but in vitro amino acid digestibility. And so that should be something that maybe not necessarily the discussion for today, but it's something that we shouldn't lose sight of, is that the same arguments will be in place for the DIAS method when you're looking at the overall adoptability, because the challenges of the primary, all of that data is right now being developed in a swine model, which is a cannulated pig model in which the cannula is placed within the end of the small intestine within a pig, and there's sampling from that.
I guess I can ask Jim. Jim, since the discussions have been around for many years, FAO 1991 report was based on data obtained in the late 70s, early 80s. Is there a reason why you think that no in vitro assay has been actually validated to date? I was trying to figure out why the hesitation to approve an in vitro method.
Yeah, it's interesting. I think, to be honest, a lot of this discussion has happened in a relatively short time period, because I think it's, if we're looking at the evolution of the protein sector, a lot of it was focused around animal-based proteins, milk-based proteins, where there was a lot of a product available, and generally there wasn't as much overarching concern about factors influencing the quality of those animal-based proteins. Soy really set the stage over the last several decades now about the fact of isolation, but soy also benefited from being a relatively high-quality protein source, and so it's less of a focus so much on that. But now that we're into an era where we're seeing many, many new protein streams coming into the marketplace, we have new cereal isolates and concentrates, new pulse isolates and concentrates, new alternative protein sources coming from, microbial protein, mycoprotein, insect protein. There's more proteins on them that are being positioned. And so we just have very limited data on that quality. And so I think it's really generated this whole interest in making sure that we've got methods to rapidly evaluate, plus the fact that most of these protein sources especially on the plant-based sector, alternative protein sector, are really tied into companies that are demanding, as we position, that they not be tested on animals. And so you sort of have that that sort of double whammy there of a tremendous increase in alternative and novel protein sources, along with the desire to reduce the use of animal testing for regulatory purposes. And so I think that's really why we're seeing so much emphasis. But I think if the timing is right, I think is really why it's taken so much time.
Thank you.
Well, this has been a great discussion, and I want to thank each of you for participating today on this and in the future on this initiative. And now we'll take questions and comments from the meeting attendees. Can you hear me?
Yes.
I'm hoping yes.
Yes.
Thanks again, Dr. House, for that excellent overview of the situation, and to our panel for the very informative discussion on considerations. At this point, we would like to open the floor for questions from the audience. I'm having some issues. Here we go. All right. First question. Is the use of enzymes of animal origin in the in vitro models a problem for vegan labeling?
This is a fascinating question. It never occurred to me until about a year ago when I was at a conference and somebody asked me that question, talking about in vitro digestibility in terms of the sourcing of the enzymes. And absolutely, most of the enzymes that we're using for in vitro digestibility are derived from animals, and I think that could pose a challenge to moving forward. That being said, there are some sources of enzymes that could be looked at, be they recombinant derived or bacterially derived enzymes, that would require further validation in terms of activity levels. So I think it is an important area, and it's something that we should be thinking about from right from the onset, so to make sure that we are really considering these as options.
Thank you. Next question for all the panelists. What existing credible, and I think this is in vitro digestibility database, are you aware of? So what existing credible in vitro digestibility database are you aware of? Are they being accepted by FDA and Health Canada?
I can start there, and Elaine, if you want to comment as well, but right now FDA and Health Canada do not accept in vitro data. There are databases being generated, and I think that is going to be critical that we make these databases open access and available for evaluation if we want to see these models and these methods being advanced. But as it stands right now, I think it would be very, very difficult to say that there is a single database available for in vitro digestibility coefficients. There are many labs generating data, and I think that that should be pulled together. The challenge, I think, is that the methods that are being used haven't yet been standardized, and I think that's really part of what we're seeking to do through the protein subcommittee is look at what would be needed to set up a standardized, validated method to the goal of establishing a database that could then be used by the sector for advancing the case, and then bringing a little bit more comfort to the use of in vitro models for substantiation of protein content claims.
Yeah, and could you hear me?
Yes.
Yeah. The CFR for the regulation on PDCAS in the Federal Register, there's also a listing there that I know the FDA does allow use of those values if your protein is equivalent to something that's already published. And then there are a few other references and selections, and actually I don't have them at my fingertips, but most of them are in review articles and stuff, but they're pretty much what's in the Federal Register if you look for PDCAS.
Okay, so then let's flip that question and ask, do you know of any existing credible in vivo digestibility databases that hold the in vivo information, and are those accepted by FDA and Health Canada?
No, that's what I was referring to, the in vivo
Okay.
data from the Federal Register. But yeah, I don't know if there's any more recent stuff, because as we indicated, very few labs are doing that, and a lot of the data was collected in the late 80s, earlier 80s.
Yeah, and certainly with the FAO has published a database. Now it's last updated, the third edition was updated, I believe, in 1981 was the last time it was updated. And that was very clearly mentioned within the 2013 FAO report for the DIAS that there's a need to update those biological databases for the protein quality.
Great, thank you. Now here's a question that is on everybody's mind, I'm sure. No other nutrient requires a digestibility score when determining the percent daily value. And in fact, the RDA, you may argue, includes digestibility already. Why would protein be different? I understand looking at amino acid scores using essential amino acid standards, but why digestibility?
It's a fair question. Certainly, I think you can. I think very clearly it's as the questioner or position, the protein content claim substantiation is the only nutrient that requires a digestibility coefficient. We actually have, built into our RDA values other nutrients that require sort of coefficients for establishing equivalence. So you have the dietary folate equivalent, which already assumes that certain proportion of folate is digestible. I think that uses a standardized coefficient. Same thing could be argued that you could do for protein, so which would obviate the need for any digestibility testing. I think coming up with an established coefficient might prove challenging, given the range of proteins that are available. I think but that is an approach that you could use a set valuation for protein digestibility. But yeah, it's a fair question to ask, certainly. And I think that's one of the reasons why moving to an in vitro testing model at least gives you some assessment of whether or not in the era of new processing methods and new blends, I think there is some merit in still having an assessment as to what extent you might be damaging or impacting the overall nutritive value of a protein source through novel processing methods.
Yeah, I have nothing to, but the last point is very relevant, particularly for food companies developing new ingredients. Minimally, they would want to have that information for in-house marketing purposes, that this is very digestible or not.
Great, thank you. The next question here, what is the ideal outcome variable that protein quality or PDCAS should measure? Is it growth, development? Is it recovery?
Well, PDCAS by definition is there to look at digestibility alone, and there has been a lot of discussion about what might be a better functional indicator of protein quality. Certainly, looking at protein synthesis rates, muscle protein synthesis rates, certainly is one option, but I think that's more of a biological research endeavor to look at, to what extent different proteins may afford better opportunities to promote protein synthesis or increase plasma amino acid concentrations. But if you're looking at strength or walking ability in an aging population, there are a number of different functional outcomes that can be looked at. Growth is certainly, for when you're looking at children, you want to make sure that the protein can support muscle growth. But I think it's a little bit of a challenging question to answer, because we're really talking about functional outcomes for protein, when in fact the PDCAS really isn't doing any of that. There's an assumption that the amino acid score is handling that piece, because you're relating the amino acid pattern relative to what is the requirement pattern. and just adjusting that by whether or not that protein actually gets digested and ultimately absorbed. So, you know, if we're still using the scoring method, I think we're assuming that the requirements have handled that biological need piece.
And furthermore, that's probably much more relevant when it's a sole source nutrition, a protein that's used. And we were focusing the discussion, as Kathy indicated at the beginning, that we're more concerned of regular consumer foods. And so it's probably less of an issue when you have a more varied protein source diet. But clearly, when you're talking about elderly or children that are malnourished, then that becomes a relevant question if it's a sole source of nutrition. But so many factors, including the gut microbiota of children, impact. You can give them a totally complete protein that in healthy children would promote growth, whereas the gut microbiota is so damaged from disease and malnutrition that it doesn't function, as some of the studies that Jeffrey Gordon here at WashU has shown.
Great, thank you both. All right, we have time for one more question, and this looks like a methodology question. Is the difference between ileal digestibility of individual amino acids different enough from the overall fecal nitrogen digestibility to have any nutritional effect on protein quality assessment?
That's a really good question. There have been some papers that have looked at comparing DIAS to PDCAS. And I think one of the biggest factors that influences the differences in the scoring is really the reference pattern, the reference amino acid requirement pattern that's used in the DIAS method, which uses an alternative, a revised amino acid pattern, and not necessarily the digestibility coefficients. So I'm saying that based on just limited number of publications. There is more data coming out. The challenge is that most of the PDCAS measurements that they're referring to are done in the same model that they're doing DIAS, which is the pig model. Whereas if you're doing the rat model, it might give you a different coefficient than relative to the pig. So there is a little bit of a lack of data to give an adequate comparison, but where there has been some published literature, I think the biggest impact has not been necessarily on the digestibility coefficient, it's actually been on the reference pattern that's been used. That's where you see the biggest shift.
Mm-hmm.
Great, very good. Thank you both. That brings us to the end of the session time. We hope everyone enjoyed learning about the considerations and rationale for measuring protein digestibility with in vitro methodology. I'd like to thank our panel and you, the audience, for some really great questions this afternoon.